Welcome to the BRAZ webinar today. It's our great pleasure to receive Henning Hermjakob. Henning is a bioinformatician working at the European Bioinformatics Institute, where he leads the molecular networks team. And today he's going to talk about Intact. Henning, thanks a lot for the availability. It's a pleasure to have you here.
Okay, thanks a lot for the chance to present here to Sandra and to Miguel. And it's a pleasure to be on the seminar series. I think it's for the second time. Probably some of you have been there for the first time. So I'll give a quick background. So the activities of my team are centered around providing network context for biomolecular data. And we do that over 4 major databases. And the first one is Intact. Intact records biomolecular interactions, mainly protein-protein interactions, but also protein-drug, protein-RNA, etc. And it's what we call a primary database. It records data directly from the literature and then records these interaction data sets in the database. And it's the focus is about interactions in the sense 2 molecules are found in close proximity in a biological system. If you then take interactions, an interaction network, and you add directionality, add things like inhibition, add additional experimental data to record reactions, which are then connecting into pathways. Then you come to pathways, which are really chains of biochemical reactions. And we record these pathways in Reactome. I spoke about Reactome about a year ago or so. And then if you take a pathway and you add a more rigid mathematical framework, you add quantitative data, rate constants, etc. for reactions, then you come to a hopefully predictive model of a biological system. And we record this kind of systems biology models in the biomodels database. All of these are connected by using the same reference systems, Uniprot usually for proteins, CEBI for chemicals, RNA Central for RNA, Ensemble for genes, etc., so that all these resources are linkable by their shared entities. One thing which does not yet exist or did not exist for a long time is a reference system for biomolecular complexes in the same way that Uniprot is a reference system for proteins or CEBI for small molecules. And so we have also developed a sister database to Intact, the complex portal, where we record functional biochemical complexes. But today I will be focusing on Intact molecular interactions. And I'll also take the opportunity to really go in the detail of how curation for a biomolecular database goes. Probably most of you are using biomolecular databases, and you are interested or less interested in the details, but I'll go quite deep in the details today, both on what is in Intact and also the rules, how we do curation because this is quite important to understand when you want to correctly interpret the data. Okay, so INTACT is a manually curated molecular interaction database. The data is not text mined or anything like this. We use text mining as a tool, but always then under human validation by PhD level scientists. It is one of the largest interaction data resources, which does its own curation with currently more than 1,100,000 binary interactions. And we follow a detailed, deep curation model. So we record not only A interacts with B, but also as much as possible, all the detail we believe is necessary for the correct interpretation of the experimental data. And that includes experimental conditions, experimental methodology, modifications of the proteins, etc. And goes all the way to, for example, the domains which are mediating the interaction if this data is available from the experimental publication. So there are a few curation principles which are quite important to make the whole thing work in the long run. And one of the important ones is that the aim of curation, database curation for us, is a faithful representation of the interaction data as it is reported in the publication. We try to avoid making judgments. There is often in interaction data a question about false positives, false negatives. Is the data reliable? In the same way it is for almost every experimental method and in almost all all domains. But we explicitly aim to record the data as it is given in the publication, rather than making a judgment on whether this is a reliable data set or not, with some exceptions. Sometimes we include, for example, author judgments, but we mark them clearly, and this can influence curation. So as an example, in large-scale experiments, authors often give different confidence levels for different parts of their large-scale data set. And so we will use this author confidence. And we also tend to not curate the low confidence section of large-scale experiments, because it has turned out that they are really just polluting the data. They are what authors call low confidence, tends to be really low confidence. Users are often not making the distinction and then are asking, why do you have such nonsense data? And so as a rule, we tend for large scale experiments not to include what is marked by the authors as low confidence data. We do make judgment calls on the publication level, whether we curate a publication or not. And this is sometimes just by by expertise of our curators or interest of curators, or whether sometimes we know from experience that publications in a certain journal tend to be difficult to curate or something like this. And so then we might just not curate publications from a specific journal. But again, in general, we try to, as faithfully as possible, record the data from a publication. And I'll mention a couple of times why. Somehow I have problems moving my slides. Okay, so one key point to keep the data compatible over time. Intex now contains data from almost 20 years of curation, and or between different organizations. I'll mention that later. There's a number of different organizations which all use the Intex database as their curation platform. and also to be able to combine the data from many different publications. We use extensive standardization. I already mentioned, we use always the same reference systems for proteins, Uniprot, for chemicals, KEBI, Brenda, for tissues, etc. So wherever possible, for almost all attributes, we use external ontologies or databases to standardize what we curate. Our curation manual, which has all the detailed rules of how we do curation, is publicly available, so that if somebody has deep questions, they can just go to the curation manual and really understand in detail how we do the curation. And this is just standard best practice in curation over most databases, and it's essential for aggregation of data from different sources. And if you're active in bioinformatics, you probably know that mapping just identifiers in the same domain from different resources is a nightmare. We use both external reference systems, and we do a lot of our own work in standardization. We have a fairly extensive set of ontologies, the so-called PSI-MI ontologies, where we define all standards for all those attributes where there are no external reference resources. For example, for experimental methodology, and there's one example for one experimental methodology given here. This is just one term which we use in the annotation, and it has a detailed description, and it's part of a fairly deep hierarchical ontology. Our work unit is always one scientific publication, and we always curate publication in full. So that means that we take all the interactions which are in this publication based on the current guidelines. We do not, let's say, if we happen to be interested currently in curating immunology, interactions which are relevant for immunology, then we do not curate from a whole series of paper only the interactions relevant for immunology. When we might pick up papers specifically which mention immunology in the abstract, obviously, but for any given paper, we aim to curate all the interactions and not do cherry picking, because it would be a nightmare to track over long term which part of a paper we have already treated and which one we haven't curated yet. And therefore, we make sure that if we touch a paper, then we curate it in full. And we have a central tracker where we record who has already curated a publication, or that somebody has reserved it for curation, so that we avoid duplication of work or conflict between different curators, which otherwise might happen easily because we are collaborating with many other international organizations. I'll come back to this. And this is all defined in quite a lot of detail in the curation manual and as part of a detailed workflow for the curation process. In a single publication, there's often multiple methods which are used to determine, often first, a large set of interactions with a high throughput technology, for example, and then a selected set of these interactions is picked by the authors for biological interest, and they confirm these then, or aim to confirm these with another methodology. And so for us, this would be two separate experiments because they use different experimental techniques. And this is for us the second work unit. So whenever a specific pair of interacting proteins is observe to interact with two different methods, then this will be for us what we call two different interaction evidences, which are generated from the same paper. And there can, of course, be even more different methodologies used in a single paper. And so we use the technical term interaction evidence, and that is for us one interacting pair observed by one technology and reported in one paper. And so one paper can result in between a single interaction evidence and tens of thousands of interaction evidences in large-scale experiments. We also try to make a good connection between the paper and the record in the database. And we will, for example, annotate from which figure the data was taken, which is recorded in a specific intact data set, intact record. And that's what I just said. So interaction evidences per publication can be between 1 and tens of thousands, which is also quite a technical challenge for the database management system to have such a broad range of interaction or experiment sizes. So why are we keeping different data from different experimental technologies strictly separate? Why don't we just aggregate everything from one paper? The key reason is that we can afterwards, across many different interaction evidences from different technologies and reported in different papers, we can aggregate the data computationally to calculate a confidence score. Protein interactions, in particular from large-scale experiments, have, especially in the early times of interactomics, suffered from a very high false positive rate. And so a major activity of interaction data resources is the calculation of confidence scores, which tell the users a kind of probability that this interaction between these two or more molecules is a real interaction and, let's say, not an experimental artifact. And there are two major approaches for this. One is machine learning. So you can calculate a score based on calibration against high confidence gold standard reference data sets, for example, interactions from X-ray crystallography. Or you can have a deterministic score where you calculate the score based on the textual annotation of the interaction. And this is what we do and what I'll focus on in the next couple of slides. So for us, the MI score is a score between 0 and 1. It's normalized to between 0 and 1, which is assigned to any pair of molecules in the database. And it's based on the number of different reports of this interacting pair, the number of different experimental technologies which have been used in these reports, and the types which are often, interaction types which are often determined from these experiments. Okay, sorry for the interruption. And so the core of this method is based on manually assigned heuristic scores for the methodology by which the interaction has been determined. And here I show a couple of typical methodologies and the scores we have assigned to them. So the higher we consider the confidence of the data from a certain experimental method, the higher the score we assign to this method, and the more this method is then uprating the MI score of that specific interaction, which has been determined by this method. And this has been published in the paper on the upper right corner. We provide detailed information in the downloads, also including the MI score, and we cluster our interactions by pair of interacting molecule, like seen on the left in the slide. Then we show the methodologies which have been used, and you can also then filter for these. I come back to this, so that you might, for example, you might think that you're not interested in anything that is derived from co-IP data, and so you can just filter this out. And then we provide the links to the publications which have which are supporting these interactions, and then the link to the detailed records in the Intex database, and then the score which is assigned to this interacting pair, often across multiple different papers which all record this interacting pair. And of course, every method needs to demonstrate that it's actually working, that it's valuable, that the scientific approach is correct. And in interactomics, among many other fields, that's very hard, because there's very few highly reliable reference gold standard data sets around against which you can benchmark any method. But we had a kind of unexpected opportunity to benchmark the method a while ago. And there was a large-scale experiment by Boxham et al., which was published in Cell, so high impact journal. And there was an erratum published 4 years later, so it took quite a while. But what happened is that due to a mistake in the data processing, actually the last third, roughly, of the reported interactions was effectively randomized simply due to an error in the spreadsheet management of the data and an off by one mistake. So all the interactions which were in the last third of the data set were more or less random. And so then we used the MI score and we could demonstrate that the MI score was much higher for the correct interactions, which were in the first 2/3 of the data set, than in the randomized interactions in the last third of the data set. And that has been then again published in the publication on the top right. So at least we are not completely wrong with this methodology. Now, apart from aggregating data into and assigning an MI score, why are we going to all the trouble of reporting all the details about the experimental methods which determine an interaction? Because clearly that costs a lot of time, and therefore a lot of money, and we could have much more content in the database in terms of different interactions if we would do a much more superficial A interacts with B method. But it's really important to understand that the methodology has a huge impact on the interpretation of the data, and we want to support this as much as possible in the database. And one example of this is a data set we created a while ago. We had specific funding from the Michael J. Fox Foundation to do a lot of targeted curation around LRRK2, a gene which is strongly implied in Parkinson's. And the aim was to, and with this funding, we could more or less fully curate the literature of experiments which reported interaction data around LRRK2, which is a highly researched molecule. And this data set contains, or contained already at the time, thousands of interactions which were reported for LARC2 and its immediate partners. And this is what you see on the picture on the left. And this on the right is the interaction data set in blue for human and in sort of pinkish for mouse, where we have filtered the huge data set on the left to only show the data which was determined under close to native experimental conditions. So with no or minimal overexpression of proteins, etc. And so that shows how important it is to really understand the context of experimental data and not just take everything, throw it all into one big database with 2 columns and then say, I have studied the interactome of my topic of interest. And that's why we really try to be as precise as possible in recording the data and in presenting it to the user. We're not alone in doing this. The Intex database at the EBI is part of the International Molecular Exchange Consortium, or short Imax, in which 12 international contributors work together for now almost 20 years. We started with defining common file formats so that the user could at least use all the data relatively easily together. But if you just have the same format and you do curation in different ways, it's not very useful. So then we moved on to working out a common curation manual, which is now in use by everybody. We had a couple of grants which allowed us an ever closer integration between the different organizations. And nowadays, all current Imax partners use the Intex database as their curation platform and their for data dissemination. But many of them also have a specific focus, have their own websites, and disseminate the data also in their own context. But we minimize the duplication of software development by having the same curation platform, and that saves a lot of money overall, because a writing system, namely the curation platform, is not very visible, but is more expensive to develop, more time cost intensive to develop than a read-only website for the user-facing front end, because you need to make sure that everything is safe, you don't write nonsense to the database, you don't have database corruption, etc. And this has been the case for now almost 10 years, that everybody uses the same curation platform. And we have had over the years a whole number of joint publications, from the definition of the joint data formats to recent publications on specific data sets we have worked on together, and I'll come back to that later. The first one of the two I want to emphasize here is our mutations data set. I've already mentioned we don't record only A interacts with B, but also a lot of experimental detail. And one key part, one key attribute which we record where about experimental detail is the effect of sequence variation or mutations. Wherever the data is available from the publication, we will record if the authors have studied both wild type sequences and mutated sequences, and we will record how the interaction has changed as so what the impact of the sequence variation is. And that is, of course, very valuable data nowadays, where it is much cheaper to do large-scale sequencing than to do large-scale interactomics. And this data set is now used extensively in the prediction of variation effects on the interactome and as a result on pathways and function. And we distinguish in the annotation between whether the variation is a natural variant or mutation or whether it's artificial. For example, systematic mutation of all the amino acids in a known binding domain. And this is all carefully recorded and accessible in the data. And this was a publication in 2019, which was the result of joint curation by 10 Imax partners over 10 years. And when we published this, there were 28,000 of these feature annotations in the data set. We keep maintaining and updating this, and by now there are 72,000 annotations. So it's a very sizable data set. A more recent key data set is the COVID-19 data set, where basically when we all went into lockdown, I'm sure you know the procedure, we started to jointly focus on the curation of interactions involving proteins from the coronavirus family. And we published this interaction data set first in bioRxiv and Twitter, and then more recently in database. And again, this is a data set which is being maintained and further developed, and currently contains more than 9,000 interactions involving proteins from different coronavirus family members, not only from SARS-CoV-2. Now, the next few slides, I'll discuss a bit how we are disseminating this data. One of the ways is the Scikit or PSI Common Query Interface. And this is an API or a query interface where many servers can provide interaction data. They all implement the same query language, which is derived from a standard query syntax and has just a few extensions, for example, which take into account the specific domain of interaction data. And one client can send the same query to a number of interaction data sources and then aggregate the data on the fly and present it to the user, and then do further things like, for example, scoring of the data based on a specific method which this particular research group might want to implement, rather than use the scoring which is provided by the source data sources. And the advantage of this compared to the many aggregating databases which are around, which just take the data from many data sources and put it together, is that it's always up to date, rather than often delayed by the release rhythm of the aggregating database. At least assuming that the source data, so the data sources all update the data in time. Another point which is somewhere between experimental detail and dissemination is negative interactions. So, of course, it's very difficult to say these two molecules do not interact. This is always a statement which is relative to the observed conditions. But if the authors have explicitly said, we have tested this pair of molecules under these conditions, and they did not interact, but they did interact in our hands with slightly modified conditions, then we will record this and we will clearly mark it as a negative interaction. We will add a flag negative in the data model, but unfortunately this tended to be overlooked by users, and we found the negative data from our data sets happily integrated in aggregated data sets and other integrative data sources, which put the data from many origins together. And so we now very clearly mark the data as negative also in the web interface. It's you can just with one click filter them out. And in the downloadable data, any negative data goes in a separate file, which has an underscore negative in the file name to make it as clear as possible that this is really different data and you should not just throw it together with everything else. Another way of dissemination is through the Reactome pathways database. I mentioned earlier on that Reactome is part of the same database family. And so we are using interactions from Intex to extend the scope and the coverage of the manually annotated Reactome pathway database. When you see the detailed molecular maps in the Reactome pathway database on many entities, you see in the upper right corner a number on a red background. If you click on this, then you get the medium and high confidence interactions from Intex. And these are also fully searchable in Reactome, so that you can, in the Reactome interface, discover an interaction, and you are then sort of led to a pathway diagram which has at least the interactor of interest interacting with a manually annotated protein, so that you get an idea of what the functionality of this protein of interest might be in a pathway context, even if it's not yet manually annotated in Reactome. And you can also overlay different data sources via the Scikit web interface onto these pathway diagrams. If you don't like the standard Intex interaction data, then you can just use another interaction data source which implements the same protocol. And this really extends the Reactome coverage. So Reactome is a manually curated pathway database, and it covers currently about 11,000 human proteins, which is a bit more than half of the human proteome. And this is extended by 5,000 interactors with medium and high confidence from Intex. So the coverage of the database really gains through the Intex interaction data. Another example of use of Intex in an upstream database is Uniprot. We have a very close collaboration with Uniprot, had it over many years. And the Uniprot interaction data content is mainly provided by the Intex database and is visible in this kind of matrix representation, as well as in the list of interactors for a given protein in the Uniprot record. And if you click on one of the points, you then get the details of this interaction and its experimental support, and you can link back to Intex to get the detailed annotation. And this is another example of where we use the detailed annotation. Uniprot export rules took years of discussion and fine-tuning, and a number of times the rules were changed of what is exported from Intex into Uniprot. And because we have the detailed annotation, we could just change the algorithm and then change what is exported. If from the beginning we had tried to manually annotate, this is a reliable interaction which is exported to Uniprot, then we would have each time to go back and recurate everything whenever we wanted to change the rules, which is basically impossible. And therefore, again, the detailed annotation is very valuable. The many pages on the right side here just illustrate the rules which determine which interactions from Intex are exported to Uniprot. So it's quite a complicated system. Now, the two most recent changes for Intex data dissemination are the Intex Cytoscape app, which we have released beginning of this year. And if you are working in the domain, you are probably familiar with Cytoscape. And we now provide in Cytoscape, through the Cytoscape app, again, access to the detailed Intex annotation. You can filter by interaction detection methods, participant methods, so the detailed experimental annotation, host organism, many other types of annotation. You can highlight any data which involves mutated sequence. This is shown in this picture by the sort of red pinkish lines. And so you get the best of Cytoscape with its many applications, with its many apps, and the detailed Intex annotation together in one package. And the most recent development, which is still not out, but should be out in a couple of weeks, is the new Intex Beta website. We have completely redeveloped the website to both provide an optical refresh, to provide a new and modern look and feel, but also to provide better interactomes views where you can get all the interaction data for a certain species more or less at one click. But again, also to expose the rich detail of the Intex interactions and their annotation, and to make this both visible and filterable through a comprehensive set of filters and visualization options. And this is, as I just mentioned, not yet released, but it's directly accessible from the Intex homepage. I'm also giving the link to the beta site at the bottom of the slide here. And we are very interested in feedback on how you like the features on the new website, what you would like to see changed, and just in general, user feedback at the moment would be really highly appreciated. I mentioned you have a very convenient access to the interactomes for a number of model species, and you get comprehensive filter methods. So if you search, for example, for this Swiss blot protein, you first get the kind of typical hairball where you can't do much with, but you can then refine your selection, for example, taking into account the specific pro chain from Uniprot. In this case, this is the specific protein from the SARS-CoV-2 polyprotein. And so you can already narrow down the search. I've also here highlighted again in red any interaction and the associated proteins which are involved, which have been observed in mutated forms. This is shown in pink if you use this flag shown on the left. I've also reduced the number of displayed interactions by increasing the confidence score with the slider shown on the top of the page. And for any and all the data which you see in the graphical representation in the main screen, you also see in a tabular representation at the bottom. And if you click on any interaction of specific interest and go to the magnifying glass symbol, you then get the full detail of just one interaction evidence as shown in this page. where you have an expanded sequence view of the partners in this interaction and all the annotated features. These are protein features, for example, experimental modifications of the protein sequence in the N or P terminus, and sequence variation, which is annotated in detail and highlighted through these color bars. And with that, I would like to acknowledge the Intex team led by Kalpana Panaselvam. Birgit Meldal is mainly working on the curation of the complex portal, but also has contributed to the data presented here. Margaret Dewsbury is a senior curator. The development team is Noemi Del Toro, Anjali Srivastava, and Elliot Ragueno. Until recently, Pablo Porras was the Intex project lead. He left because he came up to the end of his EBI 9 years. Sandra Orchard from Uniprot has been the longest together with me on the Intex team in various roles. And as I mentioned, we are collaborating with 12 contributors, or have been had 12 contributors to Intex from the Imax consortium over the years, many of them still active. And all of this has been supported over the years by funding from the US NIH, by EBI core funding, and funding from the UK BBSRC, and from Open Targets. And with that, thank you for your attention.
That's great, Henning. Thanks a lot. Great talk. Let me look at the chat here. I have a question for you. You mentioned the negative interactions.
Yes.
So it was not clear how you deal with discrepancies. So if one paper reports the interaction, the others say there's no interaction, how do you deal with that in the curation?
So again, we've tried to not make any judgments. If one paper reports an interaction and another one doesn't, then that's fine. So that is just one interaction evidence from the one paper and nothing from the other. It's perfectly possible that one paper says, reports the interaction in a positive sense, and another one explicitly marks it as a negative interaction. So as I mentioned, we only annotate this if it's really with the detailed experimental conditions, because that's essential to, you can only say something didn't happen under these specific conditions. And we then will report this negative interaction. We will mark negative with all the explicit flags as much as visible as we can make it. And then and we provide both of these data set, and it's ultimately then up to the user or to any computational integration algorithm to make a judgment how to use this data. Again, that's why I emphasized so much in this talk. It's really important to understand the details of the data and not just A interacts with B, and to be really careful in how the data is integrated. And for example, in a user interface, you might then choose to highlight this contradiction once you zoom in. As I mentioned, we are highlighting negative interactions in our interface by sort of highlighting it in red in the textual representation. And then it's ultimately up to the user looking at the data in a particular context to interpret this and draw their own conclusions. As always, the database can only highlight where to look first and how to further interpret the data. It's not providing the absolute truth.
Great. Thanks, Henning. Any other question for Henning?
I see one. Where is the tutorial? Where can I find the tutorial? So the curation, there are a couple of tutorials. So do you mean end user tutorial, which is just how to use the interface, etc., or a training course, for example, or do you mean the detailed curation manual, which I mentioned earlier? This was a question from Janilda Castro, so probably you can just add a bit of refinement in the chat box or just speak up. I don't know how you usually handle questions.
Both ways, yeah. People can write or can open up the microphone. And Henning, Miguel Ortega is here, so there he is.
Yeah, so, sorry, I could not make for the opening. I had to be away, but Henning, when you post the data, entity A interacts with entity B, do you think it's possible or it's good to have a column for modulation? For instance, P53 interacts with MGM2 and is degraded. But if it's phosphorylated, it does not interact anymore. So then modulations seems to me like so important that they could have a column. What do you think?
So we have actually we can annotate both required protein modifications. So we can annotate that a protein needs to be first phosphorylated to enable the interaction under consideration. So we can annotate that. We can also annotate resulting protein modifications. So if the result of an interaction is hello?
Somebody's calling. I don't know.
Well, that was on my side. That was on my side. Something went strange there. So somebody tried to call me. So we can also record the fact that protein modification is the result of an interaction. And so if this is discussed in the paper, we can record this. We have detailed controlled vocabularies where we can also annotate, for example, enzyme, target, etc. So yes, I agree, that's definitely valuable. But
so important that it would deserve a column there, maybe.
We have it. We do that. We do it annotated, but it's of course only a subset of the data, only when it's in the publication. And we also try to not go too much in detail there, because that is then the domain of the pathway databases, and Reactome is our sister database. And so we try to have a reasonable separation, which is not always an easy decision, between Intex as a primary database, which provides really the primary data on a per publication, per experiment basis, and then the Reactome database as a secondary database, which has a lot of human expert interpretation in drawing conclusions from, let's say, multiple experiments to, yes, this is a reaction which is a phosphorylation reaction, which is this part in a whole signal transduction chain, for example. So it's we can record this kind of data, but we try to not turn Intex into a pathway database, but work closely together with Reactome which really focuses on biochemical reactions and how they link into pathways.
It would be easier for the user to go to Reactome and to see the possible modulations of the interaction, phosphorylation, etc.
It depends what you want to see. Intex is really for more the detail on a per publication basis. Reactome is more for the larger context, how biochemical reactions form into pathways and whole signal transduction chains. So that's roughly the let's say the division of labor.
Mhm.
And going to that again, there's a question in the chat about string. So string is actually, or Intex is one of the data sources of string. And we have, well, actually very long-standing in collaboration with string. They contributed, especially in the early days, to the development of the standards which we use in the Imax collaboration, and they are using Intex as one of the major data sources for the experimental data, but of course they do a lot of prediction, they do a lot of text mining, and so they aggregate from many sources, so you can't get the detailed view, which I just discussed mainly in this talk, from string, but you get a much broader view across many more interactions, across many more species through orthology inference. But again, it depends what you want. If you want to have a quick first glance, string is definitely great to go to. If you want to go into the details underlying this quick first glance, then it would definitely be a good idea to look in the detailed Intex annotations.
I was going to ask something very related to that, but you have a network, right? You have in Intex you can observe a network of interactions.
Yes. Yeah, so that's what I showed. We have the Intex viewer, which integrates the interactions into a network, and where you also can access the same data through the Cytoscape Intex app. So we do similar things like string.
Yeah, this is very similar.
But with a different focus, not as broad as string in terms of they have many millions of interactions which are based on.
Yeah, they couple text mine interactions to build the networks. It's another approach on top of the protein interactions, right?
Yeah.
Sure. Okay. But you do not use a text mining to relate the interactions, but you have the PubMed IDs, you have the references for every interaction. I was talking to Pablo once in a meeting. We were trying to use something that we use with Adriano. Adriano working in Luxembourg, has a text mining tool, Pescador, and they wanted to use in Intact actually text mining to search the papers, interaction reported in the papers. But my question is, if I want to start with a mesh term like menstruation or psychosis, of course, if I download your database, I can go from the mesh term to the PubMed IDs and map the interactions. You do not think it would be good to have an intact such a thing. I put a mesh term there. Mesh term will bring me to the papers that will bring me to the interactions. Because it would be very nice for me to work in this way without downloading.
I agree. That is an interesting suggestion, and it shouldn't be too hard to implement. No. So I'll take that as a suggestion, and we'll see what we can do about that. That makes perfect sense to me. Basically, filtering the network or via annotation of the publication.
Exactly.
So that's a perfectly viable approach.
Also, I would add another suggestion that would be difficult for me. That would be to try to do the same with, it's more complicated, but with Go processes, gene ontology processes. For instance, I want to characterize these processes in man. Which interactions are related to the process. The good thing for you is that you are not away from the Go office there in EBI, right? Wherever you look, wherever you locate.
It's not a big deal to do it. It's
Uh-huh.
not a big deal because we can just, and we already import the Go terms which are annotated to the proteins.
Uh-huh.
So I'm not sure if that query is possible already right now, but in any case, that should be relatively straightforward.
I know, but I predicted that is not going to be straightforward because of you need some curation. For not for the mesh terms, but for the Go processes, you might need some curations. But you work with curators, and the Go curators are maybe on the next door. Do you work close to the guys there?
Well, Go is.
I know that Go is close to Nick Goldman's office.
It's a huge collaboration which is global.
I know.
Go, especially the Go annotation. So the Go offices are the offices for the Go ontology curation, which happens to some part at the EBI, while a lot of the annotation of terms. Go terms to different proteins comes from many different sources, but nevertheless, where it comes from, I think that is something which is perfectly possible. I don't think we want to curate explicitly which Go terms apply.
If you are doing a good job,
that is just a too big job. But to use existing Go annotation on proteins and then use that as a filter on the intact interaction network, that is not that is technically perfectly feasible and not that far away.
Well, I mentioned that because I knew people from the Go annotation project, and I learned that they are across the hall with the Go ontology people. So many times they did not think that the Go annotation would work well. So they just went across the corridor and asked the Go ontology people to create terms for that,
Yes. So we do that all the time, not with so much, well, with Go, I would say in not such a big scale, because it's fairly rarely that we need a Go term specifically. But we do that, for example, with KEBI all the time. If we have some kind of chemical which is not in KEBI and which we need to annotate, then we just send the request to KEBI and they create it in a couple of days for us. So that works pretty well.
Yeah. Let me see if people in the audience thought on one more question for you here.
Yeah, there is a link which I guess I will follow at some point afterwards, but I will make a note because otherwise it will be gone by the time we close the session.
Sure.
So I'll put this in my notes.
I think we're recording your conference. It's okay?
Yeah, that's perfectly fine with me, but still I would have to go back to the recording to get the...
No, I know.
I've copied the link, so that's fine. But
I just mentioned, just to make sure that's okay.
That's absolutely okay. I'm also happy I shared the talk already before. I'm perfectly happy for you to make the actual slides available to everybody. So I've got no restrictions on how this is spread. Just don't put anything in and state what that I've said it. But if you take it,
of course,
an excerpt or so, that's perfectly fine. I'm happy for the slides to be used in other.
Great. So Henning, you did not come to Salvador, but you gave 2 talks already to Brazilian bioinformaticians, right?
I hope we can catch up and do the personal meeting again in the non too far future. So I think you were not there when we started. I mentioned at the beginning of this, I'm currently in an airport hotel near Heathrow, I'm about
Sure.
to go on the first business trip again for 1.5 years. So it's getting better. Let's see.
Sure. And you see in these seminars, you have people from northeast of Brazil, from my place, from Bahia, from south of Brazil. So if you come for a workshop, we can save you some days to be in another place also, just to visit the other PhD programs, right? As we planned, you would be coming to Belo Horizonte, right, for 2 days, and then going back to the
It was a nice plan.
You know, we have an agreement signed with EBI for using a program from CAPES agency, from a Brazilian agency, that we can invite visitor professors for 15 days. It's a 15 days stay. It's a long stay, but for the visiting professors, a very short one. But in any case, we can apply for this kind of visit too. You could do join some, put some cabana visits together and visit many places. So today, probably people from some 6 to 10 states were together here in this initiative. So thanks a lot, Henning, because this kind of event adds internationalization to our programs. They appear very, very nice in our initiatives, and also the students get to know you, get to know Intact, etc. And I'm sure that's very helpful for their work. So thanks a lot for being with us. Okay.
Thank you. Thanks a lot for the opportunity to present. And thanks a lot for all the questions. And don't hesitate if you have any more questions to just send me an email or so. And looking forward to any feedback. Thanks a lot.
Okay. Thank you.
Bye-bye.
Bye-bye.